The Jordan Show on YouTube.
Welcome to the deep dive. We are really getting into some fascinating material today.
We definitely are.
Our source is a backgrounder called the CRISPR revolution, and it's all about gene editing. You know, this technology that just sounds futuristic, but it's happening right now.
Absolutely. And our mission today, looking just at the source material, is to really unpack it for you. We want to get clear on what CRISPR-Cas9 actually is, you know, how it works mechanically.
Yeah, the nuts and bolts.
Right. And then explore the, frankly, huge potential it has. We're talking medicine, agriculture, completely different field.
And the flip side too, right? The ethical stuff.
Exactly. The big ethical questions and, well, some major events that have already happened because of it. The goal is for you listening to walk away really understanding this revolutionary tool.
You got it. I mean, just think about that phrase, editing life's code. It's pretty wild. So let's just jump in. What is CRISPR-Cas9 according to this source?
Okay, so the material breaks it down. CRISPR itself, it's an acronym, stands for Clustered Regularly Interspaced Short Palindromic Repeats.
Whoa. Okay, say that again. Clustered.
Clustered Regularly Interspaced Short Palindromic Repeats. Yeah, it's a mouthful.
Definitely a mouthful.
But the key thing is, these are just sequences found in the DNA of bacteria.
Okay, in bacteria.
And the really clever part the source points out is that these sequences, along with a protein called Cas9, together they act as a defense system for the bacteria.
Like an immune system.
Pretty much. It's how bacteria fight off viruses that try to invade them.
Huh. So bacteria already had this antiviral weapon, and scientists saw that and thought.
Exactly. That's the big innovation the source talks about. Scientists didn't invent this whole thing from nothing. They saw this really precise, powerful system in bacteria and figured out how to, harness it.
To repurpose it.
Yeah, repurpose it to make very specific, targeted changes in the DNA of other organisms, plants, animals, potentially us.
Okay, that is fascinating. Using a natural defense mechanism as a super precise editing tool. So how does it actually work? How do scientists use this bacterial system to make edits, based on what we're reading?
All right, let's walk through the steps the source describes. First, scientists designed something called a guide RNA, or gRNA. It's a small RNA molecule.
Okay.
And they engineer its sequence so it perfectly matches the specific bit of DNA they want to edit.
Ah, so the gRNA is like sending it to the right address in the genome, a bit like a GPS.
Exactly like a GPS. That's a great way to put it. It guides the whole system to the right spot. So this gRNA gets combined with that Cas9 enzyme we mentioned.
The protein that acts like scissors.
Precisely. This gRNA Cas9 complex, it's then introduced into the target cells, the cells you want to change.
Okay, so the guide RNA leads Cas9 to the target. Then what? What does Cas9 do when it gets there?
That's where the action is. gRNA locks onto that matching DNA sequence, and then the Cas9 enzyme does its job. It makes a clean cut, a very precise cut right across both strands of the DNA double helix, but only at that specific spot.
A targeted break in the code.
Targeted break, yeah. Now, cells don't like having broken DNA. So as soon as that cut happens, the cell's own natural repair systems jump into action.
They try to fix the break automatically.
Right. And that repair process, that's the moment scientists can step in, according to the source.
That's the opportunity for the edit.
That's the crucial window. While the cell is trying to patch things up, scientists can provide, say, a new piece of DNA. The repair system can then use that new piece to fill the gap, effectively inserting new genetic information.
Or they could just let the cell repair imperfectly to knock out a gene. Or swap a faulty sequence for a correct one.
Exactly. They can add, delete, or replace genetic material right at the cut site. The cell's repair does the work, incorporating the change the scientists wanted.
And the reason this caused such a stir, the source says, is because it's just so much better than what came before, yeah.
That's the key point the source emphasizes. Compared to older gene editing techniques, CRISPR-Cas9 is way faster to set up.
Okay.
It's much more accurate in hitting the right DNA target, and it's generally more efficient at actually making the change happen. That combination speed, accuracy, efficiency, that's the revolution.
Okay, wow. So now that we've got a handle on the what and the how, let's talk about the really exciting stuff. The applications. I mean, with a tool like this, what are people actually doing with it? Think about how this could affect you personally. Medicine seems like the obvious place to start.
Oh, absolutely. The source material highlights huge potential in medicine. Probably the biggest area is treating genetic diseases.
Right, things you're born with.
Exactly. Conditions like sickle cell anemia, cystic fibrosis, even some types of inherited cancers. The idea is to use CRISPR to correct the underlying mutation in the DNA. Fix the root cause, not just treat symptoms.
Actually fixing the faulty gene itself, that's incredible. The source mentioned infectious diseases too.
It did, yeah. There's research going on, according to the backgrounder, looking at using CRISPR against viruses.
How would that work?
Well, the idea is to target the viral DNA that might have hidden itself inside our own cells. The source specifically mentions HIV as one virus where researchers are trying to use CRISPR to find and disable its DNA within human cells.
Okay, so fighting viruses, fixing genetic code, but the applications go way beyond just human health, right? Affecting things like our food supply, agriculture.
Definitely.
Yeah.
The source talks quite a bit about agriculture. CRISPR is being used right now to develop crops with better traits.
Like what kind of traits?
Well, the examples given are things like making plants more resistant to drought, which is obviously huge with climate change,
Sure is.
or making them naturally resistant to certain pests, potentially reducing the need for chemical pesticides, or even improving the nutritional value of the food itself.
And this works for animals too, not just crops.
That's right. The source mentions using it in livestock breeding. Things like boosting disease resistance in animals or maybe improving growth rates efficiently.
It's just amazing how versatile this single tool seems to be. The source even mentioned conservation biology, helping endangered species.
It did touch on that. Scientists are exploring if CRISPR could help endangered populations, maybe by increasing genetic diversity if it's been lost, or giving them resistance to diseases that are wiping them out. And the source also brings up something related, but maybe more complex. Gene drives.
Gene drives. Okay, that sounds potent. What did the source say about those?
Yeah, it's a CRISPR-based technology that's still very much in the research phase, but the basic idea is kind of mind-bending. It essentially rigs the rules of inheritance.
Rigs the rules.
Normally, a gene variant gets passed to about half the offspring, right? 50-50 chance. A gene drive is designed so that a specific trait is passed down to almost all offspring, generation after generation.
So it spreads really fast through a population.
Extremely fast. And the source mentions one potential application being studied is for controlling invasive species. Maybe you spread a trait that makes them sterile or only produce male offspring, drastically reducing their numbers over time.
Okay, yeah, you can see the power there, but also maybe the risks too.
Exactly.
Yeah,
Which brings us squarely to the next big section in the source material.
right. All this potential. It's truly breathtaking. Medicine, agriculture, conservation. It feels like we're getting this incredible power over biology itself. But as you said, that comes with huge responsibilities and some really difficult questions.
The source makes a big point of this. This is where things get complicated, ethically speaking.
This is definitely where we need to pause and really think through the implications. The backgrounder lays out some major ethical flashpoints. One of the absolute biggest is something called germline editing.
Germline. What does that mean specifically?
It means making genetic changes not just in, say, the lung cells of an adult with cystic fibrosis, but making edits in sperm or eggs or very early embryos.
And the reason that's such a big deal is
Because any changes made there become part of every single cell in the person who develops. And crucially, those changes can then be passed down to their children and their children's children forever.
so you're editing the human inheritance line permanently. Exactly. And the source highlights the massive concerns around that. What unforeseen health consequences could pop up generations later? Is it morally right for us to make irreversible changes to the human genome itself? Big, big questions.
Yeah, you're not just treating one person, you're potentially altering the species. And that connects to the whole designer babies idea, doesn't it?
It does, and the source tackles that directly. The fear is that with CRISPR's precision, we might go beyond treating diseases and start selecting for or trying to enhance traits that aren't related to health.
Like intelligence or looks or athletic ability, and things like that.
Precisely those kinds of things. And the source points out the intense debates this sparks. Could it lead to massive social inequality? A future where only the rich can afford genetic enhancements for their kids.
Creating genetic castes almost.
That's the fear. And it raises really deep philosophical questions too about, what makes us human. Is variation, even imperfection, valuable? The source says these debates are raging.
So it's not just medical need versus enhancement, but also who gets access, equity.
That's another critical point the source raises. There's a very real risk that these powerful technologies could just make existing inequalities worse. A gap between the genetically enhanced and everyone else. Ensuring fair access if and when these therapies become real is a huge challenge highlighted in the material.
These are definitely not easy issues. And the source brings it all down to earth by discussing a specific, highly controversial event, the He Jiankui case.
Right. That event, which the backgrounder details, really threw these ethical debates into the spotlight back in 2018. A Chinese scientist, He Jiankui, he announced he'd use CRISPR.
On human embryos.
Yes, on human embryos to edit the genes of twin girls who were then born.
And what was he trying to do? What was the claim, according to the source?
His stated goal, the source says, was to make the girls resistant to HIV infection by disabling a specific gene.
Okay. And how was that received?
With almost universal condemnation from scientists and ethicists worldwide, according to the source.
And it wasn't just general disapproval. The source listed specific reasons, right?
Yeah.
Specific violations.
Yes, the backgrounder is clear on why the outcry was so strong. There were major ethical breaches in how the whole experiment was conducted, a profound lack of transparency. Nobody really knew what was happening properly. And critically, huge concerns about the potential harm and unknown health risks for the children involved.
Right.
Were the edits even safe? Were there off-target effects?
So safety, ethics, transparency, all majorly lacking. And what happened to the scientist, Dr. He?
The source notes he was later sentenced to prison in China for his work. But beyond his personal fate, the whole incident acted as a massive wake-up call globally. It really intensified discussions about needing strict rules, regulations, international agreements for human gene editing.
It showed just how fast the science was moving, maybe faster than the ethical frameworks could keep up. So, okay, where does that leave us? Looking forward, what does the source say about the future of CRISPR?
Well, the source indicates the research certainly isn't slowing down. A big focus now is on refinement, making the CRISPR system even more precise, and crucially, reducing the risk of off-target edits.
Off-target meaning cutting the DNA in the wrong place by accident.
Exactly. Minimizing those unintended SNPs is key for safety, especially for medical uses. And the applications are broadening too, beyond just medicine and agriculture.
Like what else?
The source gives examples like using CRISPR in biofuel production, maybe engineering microbes to be more efficient. Or for environmental cleanup, think designing bacteria that can break down pollutants. And also in the wider field of synthetic biology.
Synthetic biology, building biological systems almost from scratch.
Essentially, yes.
Yeah.
Engineering new biological parts, devices, systems, or giving existing organisms totally new functions. It's a huge emerging field.
Wow, so the potential keeps expanding.
It does. And circling back to the ethics and the He Jiankui fallout, the source concludes by emphasizing something really important as this technology gets more powerful and widespread.
We need better rules.
We absolutely need robust regulations, strong ethical guidelines, probably international ones, to make sure this incredibly powerful tool is used responsibly, safely, ethically, and ideally equitably for everyone's benefit. That's the challenge the source leaves us with.
It really is a double-edged sword, isn't it? This deep dive into the source material makes that crystal clear. You've got this almost unbelievable revolutionary power to potentially cure diseases, feed the world, solve huge problems.
But yeah, as the backgrounder stresses, tied right alongside that promise are these incredibly profound ethical dilemmas. Germline editing, designer babies, access, equity. These aren't just scientific questions, they're societal, philosophical ones.
It's a tool that really forces us to ask some fundamental questions about who we are and where we're going. So thinking about everything we've unpacked from this source today, maybe here's the final thought for you, the listener, to mull over. What does it really mean for us, for humanity, now that we have the ability to directly edit our own blueprint? How do we possibly balance harnessing this immense power for good while managing its undeniable risks and ensuring that its benefits and its burdens are shared justly across the globe?